countercurrent mechanism urine concentration.
Formation of dilute urine.
When the water removal is needed, no ADS is released, so the distils and collecting tubules will not reserve water, but solute will be reserved. Waters move out with the urine with low ion solute content dilute urine. Urine osmolality will be as low as 50 millimoles per liter. In such cases, the mechanism and pathway of formations of dilute urine through the nephron. Site PCT. Amount of literate R 65 to 70%. Solute R, yes. Solute of tubular fluid isotonic 300 mmol per liter. Next, descending lymph of Hele 15% of amount of literate R. No for solute R. Result of tubular fluid hyperosmotic solutions. At the tip of the LH, the osmolarity reaches to some 1,200 mmol per liter. Next, ascending limb of loop of Henle. Amount of literate R 0%. Yes for solute R. Result of tubular fluid hyperosmotic solutions. Osmolarity 100 mmol per liter. And the last, distal tubule and collecting system. No or very small amount of water reabsorptions in the absence of ADS. Solute R, yes. Result of tubular fluid, osmolarity 50 mmol per liter.
Formation of concentrated urine. In the presence of ADH, more than 99% of water and filtrate may be reabsorbed. Antidiuretic hormone, or ADH, is released by the posterior pituitary gland to maintain homeostasis of body fluid volume. If water conservation is required, ADH stimulates the water channels in the collecting duct, allowing the water to diffuse out and the urine to become concentrated. If ADH is not secreted, the duct remains impermeable to water and the urine stays dilute. Urine concentration can be as high as 1,200 millismoles per liter with ADH or as low as 65 millismol per liter without.
Current system produces counter-current multiplier and maintain counter-current exchange rate, a hyperosmotic rental modula. The two limbs of Henle's loop are a counter-current multiplier. The two limbs of the vasa recta are counter-current exchange rate. Counter-current multiplier. Counter-current multiplication is a hypothesis describing the mechanics whereby urine is concentrated in the nephron. Initially proposed in the 1950s by Gottschalk and Mile. Definition: the repetitive reabsorption of sodium chloride by the thick ascending lobe of Henle and continued inflow of new sodium chloride from the proximal tube into the lobe of Henle is called countercurrent multiplier. Lobe of Henle as a countercurrent multiplier. The descending lobe of Henle is relatively impermeable to solute, is permeable to water. The ascending lobe of Henle is permeable to solute, is impermeable to water. Distinctive features of the tube is the sharp bend of the lobe of Henle, hairpin bend. Because of the bend, tube fluid moves downward into regions of increasing osmotic pressure. After the bend, the tube fluid moves upward through regions of decreasing osmotic pressure.
The next one we have sepsis in figure A, loop filled with isotonic fluid. The tubule initially filled with isotonic 300 millismoles per liter fluid from PCT. The next one we have figure B, pump sodium from ascending tubule. Na is pumped out of the ascending loop rising the osmotic pressure in the medullary interstitium and lower in the tubule. Note that the maximum gradient inside to out is 200 milliosmoles per liter. In figure C, we have adjusting osmolarity of descending tubule. Water flows out of the descending tubule by osmosis, rising the osmotic pressure in the descending tubule to 400 milliosmoles per liter. And figure D, more fluid flows into tubule. Fresh fluid enters from the glomerulus, pushing concentrated fluid 400 milliosmoles per liter into the ascending limb. In figure E, we have second round of NA pumping. In the second round, the NA pump produces another 200 milliosmoles per liter gradient across the membrane, but it is standing from a more concentrated solution, so the external osmolarity rise to 500 milliosmoles per liter. And lastly, we have third round of NA pumping. The third round of an NA pumping rises interstitial concentration to 700 milliosmoles per liter, and so on. In times of dehydration, collecting tubules leak urea to interstitial space, further increasing water retention by increasingly osmolarity. The role of urea: absorption of urea in the collecting tubules under the influence of ADH enhances the concentration gradient formed in the medullary interstitium.
Countercurrent exchanger. There are two special features of the renal medullary blood flow that contributes to the preservation of the high solute concentration. First, the medullary blood flow is slow, accounting for less than 5% of the total renal blood flow. This sluggish blood flow is sufficient to supply. Second, the vasa recta serves as a countercurrent exchanger, minimizing washout of solute from the medullary interstitium. The major factors that contribute to the buildup of solute concentration into the renal medullary are as follows. First, active transport of sodium ion and transport of potassium, chloride and other ions out of the thick portion of the ascending limb of the loop of Henle into the medullary interstitium. Second, active transport of ion from the collecting ducts into the medullary interstitium. Third, facilitated diffusion of large amount of urea from the inner medullary collecting ducts into the medullary interstitium. Diffusion of only small amount of water from the medullary tubules into the medullary interstitium, far less than the reabsorption of solute into the medullary interstitium.